Okay, good morning everybody. So we are here in the second day of course on the fisheries for the future professionals. Today I will be talking mainly about the ocean biogeochemistry and give you some basics of the nutrients, primary productivity, and how it regulates and why it is important. Can you go into the slide more? So we will talk about nutrients, productivity and nutrient cycle. I will talk only about few basics which I think will be helpful to you for understanding the complete process. So what you are seeing in this slide is the map of chlorophyll derived from this satellite, and you can clearly see here this is annual average. There are some zones where the productivity is quite high, and there are also zones where the productivity is very low. So in this talk we will learn about what regulates the productivity and what is required in the ocean for the productivity to take place. And first of all, we will also talk about the productivity, what productivity is. Before we go into what productivity is or how it is regulated in the ocean, I want you to give a good attention to this slide where I am showing the paleoclimatic data on the Please wait for some time, there is some technical issues which they are trying to resolve. Is it visible now? Visible now? Okay. Okay. So I was on the second slide. So here what I wanted to show you is I believe this is one of the most important graphs that we have produced in the oceanographic research as far as the biogeochemistry and the carbon cycle is concerned. In this graph what you see are two plots basically. One is the red color showing the temperature and the blue color showing the carbon concentration. And if you look at this graph very carefully, whenever the carbon concentration was low, the temperature was low, and whenever the carbon concentration was high, the temperature was high in the geological past. So what controls what? Whether this is CO2 that gives rise to the temperature or it is temperature that controls the CO2. Clear understanding is still not there, but it is largely believed that the increase in the CO2 being a greenhouse gas leads to the increase in the temperature. But the most important takeaway from this graph is that if you look at the carbon concentration over the last say 450,000 years, the concentration has never gone beyond 280 ppm. So we have mostly oscillated between 180 to 280 ppm. But what we have done in the recent 100 years is we have increased the temperature of carbon dioxide in the atmosphere from 280 ppm to say more than 420 as of now. And this is one of the reasons why we talk about the global warming, climate change, etc. And the rate of increase, if you see, has varied. In the pre-industrial era it was quite less, and as we have gone into the use of fossil fuels, petroleum, coal, the rate of increase has increased drastically, and it has consequences on the climate change. You know that the global warming, the temperature is increasing. So is it that all the carbon dioxide that we have released into the atmosphere by the fossil fuel burning or the industrial use has stayed into the atmosphere? No. 45% of the CO2 that we have emitted has only stayed in the atmosphere. So where the rest 55% has gone? They have been taken away by the ocean and on the land. Land, we know that there are plants, biota, which take this CO2 during the photosynthesis process and convert it into organic carbon. That's how we all humans survive for the food and all, for our other needs. In the ocean, we don't see them, but they are there, called phytoplankton, who do the similar process. But if you look at the biomass that is available in the ocean or in the land, there is a huge mismatch. Oceanic extent is huge, vast, but the biomass is only 1 petagram on an average. But if you look at the terrestrial plants, they contain more than 100 petagrams. But the good thing here is that the turnover time of oceanic biomass is only for a week or so. But for the land, they are stored into the organic matter in form of wood primarily, and the turnover time is more than 10 years. So though we don't see them in the ocean, they play a major role. They contribute almost equal in terms of the carbon sequestration or the photosynthetic process by which carbon is withdrawn from the atmosphere. So both are important. The land plant as well as the sea plant are important. But is it that simple in the ocean to have this photosynthesis process? Here are two main critical things. One is the availability of light. So all these processes that we are talking about on the photosynthetic process or the sequestration of the carbon takes place only in the upper layer of the ocean, which is called the photic zone. I will define what photic zone is there in the next slide. But for the time being, you assume that the layer in which the light is there in that zone only this photosynthetic process takes place. And this process is basically reaction of carbon, oxygen, carbon and water in the presence of sunlight and some minor components, which again I will discuss in the later slides, and they form organic matter and liberate O2. This is how the oxygen has come into our atmosphere. And these green dots which you see here are basically a representation of the phytoplankton. So all the phytoplankton that do photosynthesis, at some point of time they die, they sink, and they go into the deeper ocean. And it has different consequences, different roles to play. Primarily these are the sinking particles which are the primary food for the entire organism that lives in the deep sea or the below the photic zone. In the photic zone, the small fishes or the zooplankton, they eat on the phytoplankton, and those that sink below this zone, they are the food material for the deeper organic matter. Now coming to the definition of the photic zone, the light in the ocean decays exponentially, and it follows that Iz is equal to I naught e to the power minus kz, where Iz is the intensity of the light at a particular depth z, I naught is the intensity at the surface, and the k is called the attenuation factor. Now this attenuation factor is governed by many things. Some component comes from the water itself, then the other constituents present in the water like phytoplankton, debris. If you are into the coastal sea, then the organic matter, the sediment, everything contributes to this K. Depending upon the K, we can define 1 over K as the optical depth, and essentially the photic zone is 4.6 over K. In the open ocean, roughly it is around 100 meter, the photic depth. So whatever processes we are talking about the phytoplankton, it takes place only in that upper 100 meter. Now you consider the importance of this 100 meter. Whatever we talked about, the 45% contribution in the carbon sequestration comes from the ocean. This comes from the top 100 meter. So the entire biota in the ocean depends upon the whatever happens in the upper 100 meter, whatever productivity takes place in the upper 100 meter. Now is it that we keep on increasing the light or how the light plays a role in the primary production. On a rough estimate, we can define the rate of photosynthesis is production. It's not always true, and I will explain in next slide why it is not always true that rate of photosynthesis is equal to production. But rate of photosynthesis also varies with the intensity of the light. At the lower intensity, the rate of photosynthesis is low. At the higher intensity, it increases. But at some point of time, it attains a maximum. And when you normalize it with the biomass, then it is called the productivity maximum, or the assimilation number, we also say it, which is PMB, maximum productivity which can take place. If you further increase the light, then the productivity may even come down, which I am not covering here. Productivity may in fact come down, and it is called photoinhibition. The peculiarity of this graph is, the graph is defined by two basic parameters. One is called the alpha b, which is the initial slope. And by initial slope, I mean if you increase the intensity from 0 to some extent, the productivity will increase linearly. But at some point of time, it will tend to saturate. And that saturation is assimilation number. And this graph is called the PI curve. This PI curve has an importance if you want to estimate primary productivity on a regional or the global scale using the satellite derived chlorophyll data. So you need to have this PI number. PI number means alpha b, which is normalized to initial slope, normalized to biomass, and the productivity maximum also normalized to the biomass. If in a sample you have more chlorophyll, of course you will have more productivity, and if it is less chlorophyll in the water, you will have less productivity. So it is required to be normalized by the biomass to get a meaningful number. Coming back to the rate of photosynthesis, which in the last slide I was telling that rate of photosynthesis is productivity, is not exactly true. The rate of photosynthesis is of course the conversion of inorganic carbon dioxide into the organic matter, but some part of it is also consumed in terms of their own survival. So the total photosynthesis that take place, or the total consumption of CO2 that takes place, is called the gross primary production, and whatever is consumed in their own survival, in their own respiration, the gross minus the respiration is called the net primary productivity. Ultimately, what is available to the biota for the to go down or to be available for the deeper organism is the net primary productivity. Light here plays a role in a sense that at the lower height level, the productivity may not be that high, but the respiration may be a bit more. So there is something called the critical depth, a depth at which the productivity equates the respiration. So the productivity which is meaningful is basically the productivity which happens above the critical depth. And the critical depth has many impacts. For example, in the subtropical zone, it is the critical depth which defines when the bloom will come. If the mixing is deep, I hope you are aware of the mixed layer and the thermocline depths, etc. So if the mixed layer is deeper than the photic depth, then the phytoplankton will not get enough time into the sunlit layer and the productivity will come down. And once this mixed layer starts decreasing, or the water starts stratifying and the mixed layer becomes shallower than the photic depth, then only the bloom starts coming. But is it enough for the phytoplankton to do productivity in the ocean? Only the light is important? No. I will define that what all other parameters are important, but the phytoplankton plays a role in the carbon sequestration can be understood with the help of this plot. In the upper layer, the phytoplankton takes the carbon, and as they sink down, they become a food or the material for the deeper biota. At the zone below the photic zone, the decomposition of these cells or the organic matter or the phytoplankton happens, and all the parameters which are consumed in the upper ocean is converted back into their basic parameters in the deeper zone. And if you look at the carbon dioxide concentration from the surface to the deep ocean, the surface is in equilibrium with the atmosphere, but as you go deep, the concentration increases, and it is partly because the temperature in the deeper water is low, and the lower temperature water can hold more gas. So the CO2 that is converted into the deep stays there. And this process of removing the CO2 from the atmosphere is essentially called the biological pump. And the same thing which I was telling you there, the organic matter is produced into the upper layer, goes to the deep, and again they are decomposed, become a food matter for them, food matter for the higher trophic level organism. And most part of this, almost 90% of the organic matter that goes to the deep is remineralized by the bacteria and other organisms, and converted, their constituents are converted back into nutrients and CO2. So what is nutrients? This is something which is required for the ocean to take place. Now this also has a manifestation on the amount of nutrients, basically the nitrogen based nutrients, which limits the productivity in the large part of the ocean. So euphotic zone, in which the light is there, the nutrients are consumed or the nitrate is consumed. So the amount of nitrate is very low, often too low to support the productivity. In the aphotic zone or the deeper water where the light is not there, these are remineralized and all the constituents are liberated. So the deeper water has lots of N and the upper water has no N. But the deeper water has no light, so productivity can't take place. In the upper water, though the light is there, the nitrogen is not there, so productivity can't take place. In this slide, I am only trying to show the distribution of nitrate or the other nutrients with the depth. You can see that the upper layer, or say upper 100 meter or so, the nutrient concentration is very low, close to the zero. But as we go deep, the concentration increases, primarily because of remineralization. Similarly for oxygen, which again I will cover in the later part, the surface layer, since the phytoplankton are producing O2 as a byproduct of the photosynthesis, the concentration is high. Also it is in the equilibrium with the atmosphere, so and there is a regular exchange, so the concentration of oxygen is high. But as we go deep, and these are remineralized, remineralization is basically conversion of the organic matter or the reaction of the organic matter with the oxygen to produce the constituent matters, so the oxygen is consumed, and the deeper layer the oxygen is low. If you go further deep, the oxygen increases because most of the organic matter is consumed in the first few hundred meters. So the demand for the oxygen decreases with the depth, and that's why the oxygen increases slightly in the deeper layer. This is how the irradiance varies in the sea, exponentially decays, and then the zone where the photosynthesis takes place. So the most critical thing apart from the light is the presence of the nutrients. In the upper layer where the light is there, nutrient is not there. Deeper layer, the nutrients are there, but the light is not there. So we need to have some mechanism by which the nutrient comes into the surface, fills the productivity, and productivity increases, and that's how the bloom comes. This map is showing the global distribution of nitrate. In the tropical or the equatorial belt, particularly in the Indian Ocean, you can clearly see that the nitrate concentration is very low. But there are zones in the ocean, for example, the Southern Ocean or the Arctic Ocean, where the nutrient concentration, at least the nitrate concentration, is quite high. Nutrient does not mean only nitrate. There are other constituents which also are defined as a nutrient. Now, why this distribution is there, that some part it is low and some part it is low, or some part it is high, is partly governed by the circulation pattern. In the Southern Ocean, the mixed layer is quite deep, so the nutrients are there, but the light is not there. So nutrients are not able to get consumed by the phytoplankton because of the light limitation. There are other limitations, for example, the availability of iron. You must have heard about this. Professor Martin from the Hawaiian Institute has once proposed that give me half a tankard of iron and I will give you ICES. He had showed through experiments that in these zones where the nutrient concentration is quite high, the equatorial Pacific where the upwelling takes place, and in the Southern Ocean, so much unutilized nutrients are there that if you add the iron, which is not there in the good enough amount to support productivity, If you add iron into these areas, all the nutrients will get consumed, and in this process CO2 will also get consumed from the atmosphere. The atmospheric CO2 will come down, and will come down to such an extent that it will give rise to ices. Later on it was found that this is not true in totality, but we cannot negate the fact that the limitation of iron is a fact in this part of the ocean. So what are the major nutrients phytoplankton need? Light is there in the upper water column, CO2 is there in the nutrient, CO2 is there in the water column, water is of course there because we are talking about the ocean. What is not there in the water, in the upper layer, are some key nutrients which are required for the photosynthesis to take place. And these nutrients are nitrogen, which comes in many forms, nitrate, ammonium, urea. Dissolved organic nitrogen are also a source of nutrients for phytoplankton. Phosphorus, PO4-3 is the another nutrient, and then silicate. Many organisms need silicate. For example, the diatom, whose skull is or the body is made up of silicate. If the silicate is not there in the water in the enough concentration, then the productivity will not take place. And the ratio in which it is required in the ocean, people have observed through analysis of the water as well as the phytoplankton that the ratio is nearly fixed, which is called the Redfield ratio. C is to N is to P is 106 is to 16 is to 1. Silicate is also required in the equal amount, so N is to silicate is almost 1 is to 1. So any of them, if it is not there in the water column, no matter what the other constituents and how much they are available, productivity will not take place. Now from where this thing came? As I said, people observed the concentration of all these nutrients in the water, and they also measured their concentration in the phytoplankton cell, and they came up with the equation that 106 CO2 plus 16 NHO3, basically 106 moles of CO2, 16 moles of nitrate, 1 mole of PO4, and plus some water will give you organic matter which has this following composition. And the major byproduct is the oxygen. This is how the oxygen is produced in the nature, and this is the oxygen that we use for our own respiration. Is it fixed everywhere, the Redfield ratio? There are cases where there is deviation in the Redfield ratio, but usually it is N is to P is close or equal to 16. But if there is a process called nitrogen fixation, there are some phytoplankton in the ocean, they do not depend upon the dissolved form of the nitrogen, for example nitrate and all. They can directly fix N2, which is 78% in the atmosphere. They can directly convert this nitrogen, they can assimilate this N2 into their body and do the productivity. In such case, the demand of phosphorus is less, and hence the N is to P ratio increases to more than 16. There are also cases where the demyelination rate affects the N is to P ratio. For example, the process called denitrification, I will explain later. In the process of denitrification, then in the absence of oxygen, nitrogen constituents are converted back into N2. So we lose N2 from the ocean. In such a case, the N is to P ratio will decrease. Now, what are the sources of the nutrients? As I said, the deeper water has more nutrients or the nitrate. So by some mechanism, if this deeper water are brought to the surface, they will bring nutrients along with them and fuel the productivity. One of the major process for this is the upwelling. Upwelling is the transport of the deeper cold water to the surface. And when this upwelling takes place, lots of nutrient comes into the surface. The another process is called convective mixing, which usually happens during the winter time in the subtropical as well as in the Arabian Sea. So during the convective mixing, the mixed layer becomes deep, and it helps transport of nutrients from the deeper layer to the surface. And then we often see the bloom. For example, in the winter monsoon, during the later phase of winter monsoon, we see large bloom of phytoplankton in the northern Arabian Sea. So depending upon the source of this nitrate, we can divide productivity into two parts: new production and regenerated production. New production is that part of the production which is fueled by the transport of the nutrient into the surface layer. Now other constituent may also be a part of new production, for example N2 fixation, but it is poorly estimated in the global ocean, and the fraction compared to the productivity that is supported by the upwelling or the convective mixing process, the productivity by the N2 fixation is very low. So it is often not accounted for. So the major amount of new production comes from the transport of nitrate into the surface layer. Now previously we have seen that the organic matter which is produced into the surface, some part of it is getting transported to the deep, but a major part is also getting recycled into the photic zone itself. So in the recycling process, the organic matter is converted back to their original constituents, and when they decay, they liberate nitrogen in the form of ammonia and urea. So whatever productivity is supported by the uptake of these nutrients are called regenerated production, and the sum total of new production plus regenerated production is called total production. Using these two definitions, we can also define a parameter called F ratio, which is ratio of the new production by the total production. And if you have a productivity map, for example using PI curve, if you can convert the satellite chlorophyll map into the productivity map, and you have this F ratio number, then you can directly estimate how much of it will go down, because whatever is going down in the deep will only constitute to whatever is comes up if you integrate over a longer time scale. Otherwise system will not be survive. If you keep on only pumping the nutrient from the deep to the surface and nothing goes down, at some point of time there will not be enough nitrate into the deeper layer. At this same time, if you regenerate all the productivity that takes place in the photic zone and nothing goes down, then again in future you will have nothing in the surface layer to support the productivity. So new production in the longer time scale gives an estimation of the export production. This I think we have already covered. Coming back to the oxygen, the major source of oxygen into the atmosphere in the geological past was through photosynthesis, but now we have enough oxygen into the surface, into the atmosphere, and the upper water column constantly exchange oxygen with the ocean and the atmosphere. So the surface layer is often saturated, we say, in terms of oxygen. If there is more productivity, for example in the bloom case where the productivity is more, you may in fact have more than 100% oxygen in the upper water column. But this is only true for the upper water column which is exposed to the atmosphere. Once you start going deep, there is no source of oxygen because the exchange takes place only in the upper layer. There is no photosynthesis in the deep, but there is a lot of respiration in the deeper water column. Respiration means the organic matter is getting remineralized. The living organism take oxygen from the water column, so the oxygen concentration decreases in the deeper water, and we have something called the oxygen depleted or the oxygen minimum zone in the subsurface layer. What are the zones where the oxygen concentration is low? In this map you can see the blue color zones are the zones primarily where the oxygen concentration is very low. They are in the Arabian Sea, the Bay of Bengal, in the tropical North and South Pacific Ocean. The oxygen is so low that it becomes close to zero at some level, and then we say it as the anoxic zone. In the Arabian Sea and the Bay of Bengal, both the basins have low oxygen, but the concentration of the oxygen is much lower in the Arabian Sea compared to the Bay of Bengal. The difference may be very small. For example, in this lower right panel graph, you can see the difference at the mid layer between say 200 meter to 400 meters. The oxygen concentration I have zoomed in, and the red one is the Arabian Sea, which is very close to zero, and the blue one in the Bay of Bengal slightly above zero. But this slightly above zero makes a huge difference in terms of the biogeochemical process such as denitrification. So depending upon the amount of oxygen that is present in the water column. The different zones of the water column can be defined as oxic zone, which is basically the upper layer where the oxygen is abundant. Hypoxic zone, where the concentration of oxygen starts decreasing or it is low on the approximately 60 micromole per kg. Then there is suboxic zone where it is quite low, and then the anoxic zone where the oxygen is not there. What happens when the oxygen is not there in the water column? And why it is important for us to know what will happen to the oxygen in the future? Because of the process called denitrification. Denitrification is the canonical process where once the oxygen goes below zero or close to zero, but the organism living there need oxygen. So once the dissolved oxygen is finished, they start looking for the other compound which has more oxygen. For example, nitrate. Nitrate has the most oxygen apart from the dissolved oxygen. So the bacteria and all starts withdrawing oxygen from this nitrate. So the nitrate is slowly getting reduced to from NO3 minus to NO2 to NO to N2O and finally N2. Here two things we have to pay attention to. One is that the production of N2O, which is potentially more harmful in terms of the greenhouse gas effect compared to CO2, and the production of N2, which is a gas, and once it is converted to N2, it may go back to the atmosphere. So ocean is losing N. At one hand, the oxygen, the surface layer does not have enough oxygen or enough nitrate for the productivity to support, and on the other hand, through this process, the N2 is being lost into the atmosphere. So if there is an imbalance in this process, then it may hamper the productivity on a longer time scale. There are other small processes also which affects this nitrogen content in the ocean, and the important of them is the NMOX, which is basically the ammonium anoxic oxidation. So in this process of NMOX, the 1 mole of NH4 and 1 mole of NH2 minus combine together to directly give N2. So this is also have been observed, and some recent work shows that in many basins the NMOX is more dominant than the denitrification. Luckily in the Arabian Sea, we have the NMOX is not that significant. How it affects the Redfield ratio? The Redfield ratio is NH2P is 16. So this black line is showing that typical NH2 and NH2P is 16:1 ratio. If you plot the global data, then this is the kind of distribution that you get, and it tells us about two things primarily. First is there are zones where the N2 fixation is dominant, and here you see more N and less phosphorus because nitrogen is getting provided into the system. But the major chunk comes from the denitrification where we are losing N and not P, so the N15 ratio is skewed. We can also define a parameter called N star, which is N minus 16P, which will give you an idea about the nitrogen deficit in the ocean. And it is very helpful to estimate which zone is denitrifying and which zone is N2 fixing. Why we should worry about the oxygen? Because of this denitrification, if the oxygen concentration, which is already low in some part of the zone, the global ocean, if it goes further down, then it can enhance the denitrification. We can lose more N, and also the fisheries, all the organisms which live in the deeper water, if the oxygen is not there, they will not survive. And there are incidences that the coastal water particularly has increasingly become anoxic because of the lots of nitrate input into the coastal waters, and the nitrogen components which are used as fertilizers are being washed away into the coastal sea. The coastal sea becomes productive, and this productivity, when it sinks down, consumes more oxygen, and oxygen becomes close to zero, and we call it a dead zone where the organisms cannot survive. So if you don't do something to keep an eye on it or to stop it, slowly there are enough evidences to suggest that the oxygen depletion is already taking place in the global water. There is another reason for the oxygen depletion in the oceanic water, and that reason is the increasing temperature of the water. The colder water can have more oxygen. So if you increase the temperature of the water, then the holding capacity of oxygen will go down. So global warming will have a negative impact on the oxygen concentration. Here I am summarizing what I have said, that global warming will lead to surface heating. Surface heating will lead to increased stratification. Increased stratification means there is no exchange of water with the atmosphere, no exchange of oxygen. So the residence time of the deeper water will increase, and if they stay there for the longer time and the organic matter is coming down, more oxygen will be consumed, so oxygen will start depleting. The increased stratification also means less upwelling, less productivity, less export production, and less demand for the oxygen consumption. So the final end product is the balance of the both, and we need to have good measurements to come to a conclusion that what is more dominant, whether the increased stratification has more role to play in the oxygen, or the less upwelling, less demand is more important for the inventory of oxygen in the deeper water. That's all for now. We will cover in more detail in the advanced course. If you have any question, you can ask. One question is why nitrate is always high in the fresh water than in the marine waters. And one obvious reason I see is that lots of fertilizers that we use is being washed away from the fresh water to the sea. So there are more. Also the fresh water is more turbid. For example, river and all, if you see, they are more turbid, not that stagnant. And then the species present there don't get enough time for the production. So the nitrogen, whatever is there, may also be underutilized. Second question is, does upwelling activity contribute in enriching the water with nitrates? Yes. They are the primary source of nitrate into the surface layer. For example, the Western Arabian Sea, there are intense upwelling during the summer monsoon, also along the Kerala coast. And you see that the productivity during the summer monsoon in the Western Arabian Sea is quite high, primarily because of this new nitrate input into this surface layer. So yes, upwelling provides nitrate into the surface layer. How can we measure the BOD in water using satellite? I don't think we have, as far as my knowledge goes, we have any technique to estimate BOD or in fact oxygen from the satellite. You can only get some information from the satellite. For example, the depth of thermocline is closely correlated with the depth of oxycline, and depth of thermocline is also closely correlated with the sea level anomaly, and sea level anomaly you can get from the satellite. So once you have sea level anomaly from satellite, you can use the relation between sea level anomaly and thermocline to get an idea about thermocline, and once you have an idea about thermocline, you can use the relationship between thermocline and the oxycline to get the information about the depth below which the oxygen is low. That's all I guess you can get from the satellite data. Thank you. Okay, Since they are taking time to stop, I will take another question. Is temperature iron having any correlation? I guess no. There are regions in the world where the iron concentration is low. Actually, people were puzzled to see that why, despite having large amount of nitrate in the polar ocean or the southern ocean, the productivity is not that high, and why they are unutilized in that zone. So earlier many hypotheses were given. One hypothesis was about the unavailability of the sunlight. As we go to the polar ocean, the intensity of sunlight increases, so they are not able to use it up optimally. Second hypothesis was about the deep mixed layer. The mixed layer in the polar ocean is often quite deep, 300-400 meters, and the deep mixed layer and the photic zone is very shallow, so phytoplankton will not get enough time in the sunlit layer. Sunlit layer itself has the intensity of sunlight is low, so both combined together the productivity was very low, and so the nitrate became unutilized. But Dr. Martin measured dissolved iron